So he's already mad. So like, coach was already mad, and then I texted him and said, dude, like, even it doesn't matter. I can tell. Practice, we never do. No, we ain't have enough guys. And he was like, fucking one text. I know.
All right, it is class time, so we will go ahead and get started. I anticipated a pretty small class today, being the Tuesday before you all have to leave. And likely this is your last class of home. How many of you this is your last class today? Pretty much everybody. All right, well, hopefully it's not too painful to sit through one more class. So I handed back your exams. Overall, yes, it was a lower average, right? I think a combination of two things. First of all, the content was just a little hard, right, at this point. And I think we're all in kind of a mid-semester slump, which I think is probably the greatest factor here as to why we probably had a lower average. We're all very tired, and we are in need of a Thanksgiving break. I did post test corrections. But there are not many, right? So overall, you either did well or you did not do well. There wasn't, in fact, there was only one question that consistently everybody got wrong that I felt like was probably an error on my end. But everything else was really hit or miss. There wasn't like one question that stood out to me that everybody just got wrong except for that one. So as we head into thinking about the final in a couple of weeks here, right? The final's in what? After we come back, 2.5 weeks until the final. Start thinking about what went well, what you might want to study more on. I will give you more guidelines on the semi-cumulative part of the exam once we get back to break, but I will give you basically a study guide of what you should focus on for the semi-cumulative material. Test corrections. Right? This is going to be in a different format than I've done it before. Right? This time around, I have linked a document up on Moodle. I want you to type in your answers, print out that document, staple it to your exam, and turn it in. It's not due until the Thursday after we get back, simply because I anticipated many students will not be here today to get their exam. And it's only really one question or a couple of questions that are paired together. Right? 8B and 8C, if you correct it, fill out that sheet, you will get full credit back for those questions. Right? This was one where almost everybody got it wrong to the point where something went wrong on my end. Right? So I wanted to make sure to give you a chance to get the points back there. Now it is only one, because I did see a lot of evidence of cheating in this exam. Maybe not from you. But there was a lot of evidence to the point where I only wanted to give back the question that I felt was bad on my end, right? So, if you have any concerns about that, feel free to chat with me. Otherwise, we will move on. Like I said, either if you studied, you either did really well or you didn't do well. There is a little bit of in between, but it's either you did good or you didn't. So I think it's a matter of that mid-semester school. All right. We're going to move on to content then. Content today, and we'll get to that content check before we leave class. And we're going to continue on what we talked about last Thursday. We started talking about Mendelian genetics and those Punnett squares. And we're going to start class today with a concept that you should be familiar with, because we talked about it last unit. All right, you should be concept with the principle of independent assortment. So Gregor Mendel, right, our pioneer in this field of genetics, working with his pea plants here, came up with these two different principles. Right? He came up with the independent assortment principle, which is how those chromosomes randomly align on that metaphase plate. right? They can align in a variety of ways. In humans, we have 23 chromosomes. There's over 8 million different possibilities of how they could arrange on that metaphase plate, right? This should not be new information. So I'm gonna move to the next slide that has my image here, right? Just kind of demonstrating this, get our brains flowing, thinking about independent assortment, right? So we're talking about in meiosis on the metaphase plate. Chromosomes could randomly align in one arrangement, or they can align in a different arrangement. And that's completely random, and that is independent from each other. All right, like I said, should not be a new concept. The other principle of that Mendel came up with is the law of segregation. Right? And this one is a new concept that we haven't talked about yet. So the principle of segregation, or the law of segregation, is that gametes have to have, let me restart here. Alleles have to separate before they end up in a gamete, right? So if we're thinking about humans, we have two different alleles, right? One from mom, one from dad. When we go through meiosis, they have to split apart, so they're going to end up in different gametes. All right. So if we're thinking about setting up a Punnett square, right? This is why we set up a Punnett square the way that we do, right? If we're looking at one allele, in this case we're looking at pea plants, right? And pea seed color, right? They both have the same genotypes, right? Capital Y, lowercase y. So two different versions of the allele. When this individual goes through meiosis to pass down these alleles have to split, right? Either it's going to pass down the capital Y, or it's going to pass down the lowercase y. It's not going to pass down both. It has to split. And if we're thinking about splitting these alleles, we have to think back to last unit, to the stages of meiosis, right? These are going to split during anaphase. All right. Same if we look at these 3 pairs of chromosomes here, right? Ultimately, they're going to split so that each gamete only has one possible version or one allele. All right, so we're going to kind of focus on this law on the next few slides and how this law can be violated in biology. All right, so far, this law of segregation, the principle of segregation, right? When we have talked about meiosis, right? When we've talked about separating out genotypes, when we've talked about setting up Punnett squares, we have talked about genotypes and phenotypes coming out in a very specific ratio. In fact, if we were to look at phenotypic ratios off of a line of true breeding parents, so remember true breeding parents, they still have 2 alleles, but they're the same alleles, right? They make that F1 offspring, which is the hybrid. The F2 generation after that is crossing 2 of those hybrids. And if we look at that F2 generation, we get that very distinct ratio of phenotypes of 3:1, right? And Mendel saw this many, many times. Right, we talked about how he saw this in every single one of his traits, from seed color to peat pod color to stem length to flower and pod position. He saw this ratio of 3:1. All right, now when he saw this ratio of 3:1, he was only looking at one trait at a time. All right, before we move on, I wanted to do just a quick review to make sure we're all up to speed on our Punnett squares, which we should be, but this is a fun activity nonetheless, right? I want us to do a group review. We're working with somebody next to you, or if you feel uncomfortable working with someone next to you, you can work with this individual with the traits shown on the screens. Determine your phenotype, your partner's phenotype, as well as your possible genotypes for your phenotypes. And then choosing 2 of those traits, create a Punnett square for what your possible offspring would have. And think about phenotypic ratios and genotypic ratios. All right, so for example, I did myself compared to this individual for the hairline, right? I have a straight hairline. This individual has a widow's peak, right? I determined our possible genotypes, right? The partner's possible genotype could be one of either. I chose one of those genotypes to do a Punnett square to determine my possible offspring's combination with this individual. All right, we'll do this for about 5 minutes here.
Then you have the two of them. It's like this. I don't know. Did you remember this stuff from high school? Really? It took a while, like my junior year. Really? I think mine was my junior year. Oh, really? Bio. No, I just had to call it a bio. Oh, really? Yeah, it's gonna hop in. Yeah, like this is the stuff that I actually, like, out of all the bio stuff that I did, this is the stuff that I actually remember. Really? Maybe it's just because I enjoyed it much first, like, some about, literally, it's like math. You know, like, if you
It's like extending a little bit. Mine's very straight up and down. I definitely don't have pictures. Always variations to these stories too. Caitlin, do we have hitchhiker's thumb? Yes. A little bit.
I think you just straight. Oh, so I'm recessive.
Wait, I don't know how to do this.
No, I didn't say it's a lot.
Right, I like it.
That's what I heard. It doesn't look like it's coming till tonight.
Gorgeous scientific illustration.
It said like 7.
Yeah. We currently have one.
Yeah,
He's a senior right now. Which is great. Yeah. Yes. Oh, it's fun deal.
Yeah. I've drawn brains for it, but I think
It's supposed to rain all day, but it's not supposed to snow until like 7.
do you like this?
It's gonna rain and then it's gonna be so fun.
Yeah.
Yeah, it looks like
Nice driving.
Or do we have it? It's hard we have longer hair.
Shitty unfortunately.
No, you're the listener.
I did those.
Oh, no, they're way more complicated.
That's how you did it, but after spin. Yeah.
It wouldn't be, but like in a perfect world. Actually,
I think wheels peak, absolutely. Got me messed up. I feel like that just can't be real. Like, imagine it all looks like that.
It is.
Bro, I know it's real, but
High
like,
school friends had one, and he used to only get on Wisconsin.
I feel like at that point you just play like the cover of your Have you ever seen what they look like when people cut them? Yeah. It's so bad. I don't even know how you look that for those people.
It's cold. It's cold. I actually haven't looked that up.
No, it doesn't even look that bad.
Now I'm serious.
I think I just turned off. I think I just get a
Yeah.
like this.
Yes. That was like I swear.
Like that just so like both ways. Yeah, like he's got like a mark down his head. It's swallowing him.
Right. Maybe this tree.
Like that's what that looks like.
Oh, okay, okay. That's a
I
non-problem.
like they lined him up, but they left just a little bit extra.
Yeah.
Why would they leave that, bro?
Like that. They try to cut it, but
I would say you're that.
or if you look at the mohawks, it's gonna cover it up.
All right. How'd it go? How many of us have a hitchhiker's thumb? A few. Yeah. All right. I definitely don't. Mine does not hyperextend at all. All right. Let's see. So hopefully you went through and you determined all your traits. Did anybody get an instance where you and your partner's combined genotypes led to 100% of that phenotype showing up? Yeah. Had at least all a dominant allele in every single genotype, or they were all recessive? Yeah, yeah. This is one's hard to review as a class because everybody's answers are different. I will say, right, for these different phenotypes, this is very simplified. In all reality, there's a lot more that goes on to determine whether or not your hairline is straight or has a peak than just one allele, right? But we have very much simplified it for this exercise. All right, any questions before we move on? All right, we're going to start talking about more complicated Punnett squares. Because now instead of talking about one allele at a time, we're going to talk about two alleles at a time, or two different traits, I should say, at a time. So we're going to look at two-factor crosses now, right? Now, two-factor crosses is going to follow two traits at the same time and how they are inherited, right? So maybe it's you crossed a pea plant that bred true for two characteristics. Maybe it was color and pod shape, right? But you're looking at two at once. Now, when you're going through these two-factor crosses, right, there's two possible outcomes that you can see when we're crossing two true breeding parents. We're looking at the F2 generation of a true breeding line. Either we're going to see that these two traits are linked or they're independent. Right? So we're leading back to that principle of that law of independence, or independent assortment. Right? If they're linked, they're always going to be inherited as a unit. Right? So if you're passing down allele A, you're always going to pass along allele B with it. Right? They're going to be linked together. They're not going to split apart. They are not going to independently assort. Versus if they are independent, right? That's kind of what we've been talking about with our one-factor crosses. They will independently assort. How one allele splits does not determine how another allele is going to split during anaphase. Right? Now there's a reason why these alleles could be linked, right? And the biggest reason is that they are on the same chromosome and they are close together on the chromosome. So if we look at this top pair, right, we have two different chromosomes here. And we have two different genes, gene 1 and gene 2. They are on opposite chromosomes, right? When these genes are passed down, they are not going to be linked together. They should be passed down independently, right? They're going to independently assort and follow that law. But if it's on the same chromosome, particularly if it's really close together on the same chromosome, then they're not going to independently assort. Then they're always going to be passed down together as a pair. They will be linked. All right, so my next slide just kind of reiterates this concept. All right, so they're physically associated with each other. Right, we call this that they are linked or they have linkage. And again, linked genes, they're always going to be passed down together. And it's most likely due that they are on the same chromosome and they are very close together. We could have instances of having the genes on the same chromosome, but they're further apart so that they are not linked. Right, now the reason for this, why closeness matters, has to do with crossing over in prophase. Right? When crossing over happens, if these genes are really close together, that chunk that breaks off of one chromosome and moves to another chromosome is likely still going to contain those two genes, right? Because they're so close to one another. Versus, say on this example on our slide here, if just the lower half of this chromosome was exchanged with another chromosome, it's only exchanging the one gene because the other gene is pretty far away. All right, so we have two possible outcomes with our two-factor crosses. Either they're linked or they are not. All right, so if we look at examples of how this would play out, right, if we're going through the process of meiosis, we're going to make Punnett squares for both examples, right? Let's look at these linked genes first, right? We have our true breeding pairs. So each parent only has one type of allele, but two copies of it. They're making their hybrid F1 generation. This F1 generation is bred together to create our F2 generation. Now notice how the alleles are passed down, right? If there is a dominant Y, it's always going to be passed down with the dominant R, because those two genes are linked together, right? They're likely really close to each other on the same chromosome. Same with the recessive alleles, right? Those recessive alleles, if a recessive Y is passed down, a recessive R is going to be passed down, right? Because they are linked. We are not going to see an instance of a capital Y and a lowercase y, right? That will not happen because of this linkage, because they are connected to one another, right? Crossing over is not going to lead to more variation, right? We're just swapping what chromosome it's on. All right, so think about this in terms of a homologous pair here, right? Now, if we had an unlinked two-factor cross, right, we would see many more combinations possible. Because crossing over could occur, that could create these gametes with much more genetic diversity to them. So again, we start with the same parents, we get the same F1 generation, but the F2 generation, because they are not linked, we can now get heterozygous individuals, right? We can get a capital Y and a lowercase r, or a capital R and a lowercase y, right? They are not linked, they're sorting independently, right? So we have many more different combinations. And again, this is likely they're far apart on the same chromosome, or they're located on different chromosomes. All right. I want to make sure I'm not going too fast here. All right, so we know we have these two possibilities. They're either linked or they're not linked. Well, what are the outcomes of a two-factor cross? Right, and this is a two-factor cross that Mendel did. Yellow round seeds crossed with an individual with green wrinkled seeds, right, produced the same F1 generation, yellow round seeds. That F1 generation was self-pollinated or crossed together to create an F2 generation with all the possible phenotypes, right? And when he did this, he noticed a certain ratio appearing, just like we saw in a one-factor cross, where we always saw a phenotypic ratio of 3 to 1. Now in a two-factor cross, he saw this very specific ratio of a 9 to 3 to 3 to 1 ratio, right? So when this happens, when we get that very specific 9 to 3 to 3 to 1 ratio, and this happened quite often, right, we learned that they're assorting independently, right? They should be following this ratio. This is a ratio we should expect if they're segregating and following the law of segregation appropriately. All right, so this is one of our outcomes. This is the outcome where they are not linked. All right, so let's go through a two-factor cross, two-factor Punnett square, where they are not linked, right? So we're gonna cross two heterozygous individuals, heterozygous for two traits, right? So both individuals will have this genotype that I've shown on the screen. The first thing we have to do is figure out all possible gamete combinations that this individual could have and potentially pass down, right? What gamete combinations could arise through meiosis? All right, so the way I like to do that is take the first letter for my first allele and start matching it with my second allele, right? So I would combine big R with big Y. That's a gamete possibility. I could also combine big R with little y. That's also a gamete possibility. All right, I've exhausted my combinations with my capital R. I will go to my lowercase r now. Right, lowercase r, capital Y, or both lowercase letters. Okay. These would be all the possible combinations that we could get after meiosis of a gamete that this individual created. Right? I would then put all those possible combinations for both individuals across the top and across the side. Now notice instead of a Punnett square with 2 columns and 2 rows, now we have 4 columns and 4 rows, right? Because we have 4 possibilities of gametes for the individual with this genotype. Right, now once we've figured out the possible gamete combinations and we've lined it up across the sides, it follows the same Punnett square rules. We just match them together to get the possible genotypes. Right? And because this one's independently assorting, and because we're following a true breeding line, we see that 9 to 3 to 3 to 1 ratio appearing. Right? Telling us once again, we know this is independently assorting. All right. Best way to learn this is to practice it. So let's do a practice problem. This is not a true breeding line, so we're not going to expect any certain ratio to appear. We are going to cross an individual that is a butterfly that is red with a striped body, with a butterfly that is yellow with a dotted body. I have given you the genotypes for the parents. You'll have to figure out the possible gamete combinations and then fill out the Punnett square. I'll give you 5 minutes to do so here. Have to head out. Yeah.
Thank you.
Yeah, no worries.
Have a good break.
You too. Yes. You still pick the
Punnett squares? But with 2, yeah, really two, what is it? What are they called? Alleles? Yeah. No, wait. 2 traits. Two genes. Yeah. Brain?
All of the folds are accurate.
What?
All of the folds are accurate.
Yeah, why not just tell?
Amanda.
It was annoying to do.
What class is it for?
It's not for a class, it's for neurology journal cover contest.
Oh, wow, that's impressive. I'm like,
If it was for an art class, it would be this one.
that is a
Yeah, see, but the accuracy
his parents figure out. That is so impressive.
All that is right.
I guess.
So how's that feeling?
I sure hope you don't quiz me on the parts of the brain with that.
Where's Broca's area? Where's
Okay,
Broca's
that I know.
area?
That I know. I know the general where it is.
I think we have 2 more candidates coming in the next couple of weeks.
I know, like, do you know where, like, the Sylvian fissure, the Sylvian fissure thing?
They're always
Isn't that the midsection?
Yeah, and then there's the,
Along that line.
I think what I'm having right now is the absolute refractory period after a period of being able to be productive. I'm not, how would you?
Yeah, but all you're saying is
Yeah.
based off of.
Also, it's like
Yeah.
What?
It's like almost great.
Thank God.
Yeah, I'm excited.
Yes. I mean,
I echo your sentiment.
So dominant is red.
you know what I'm talking about, right? With the absolute refractory period?
Yeah.
Okay, good. I'm like, you probably know that.
So do we just like list
Yeah, so like big R.
So you do RRs. Unfortunately, my absolute refractory period doesn't seem to be serving as like a function as voltage gated sodium channels.
I think so.
Unfortunate.
Is your second cube big R, little R, little S? Wait, sorry, second cube. Big R, little
I
R.
wish. Like, just New York that only half the people.
Oh yeah, most people just were like,
I'm going home early. The perils of having the Tuesday before break class.
Well, okay, this is the first class I have this Tuesday. I have 2 more today.
Okay.
So,
Okay, so this is not your last class. Lots of times Tuesdays are like lab days, so on a break week, you don't have labs.
real happy about that, but then I was also confused because I moved my OCD routine to yesterday, and also yesterday, we had mass rehearsal on Monday
Okay.
evening instead of Sunday evening, so I kept thinking today is Monday.
Yeah, that'll throw you off.
Because there's 2 things I do exclusively on Sundays, and I'm like, that I shoved, both got moved to Monday, and I'm like,
Yep.
something's wrong.
I work on Sundays, so I could never get picked.
That's fair. It's weird because I actually haven't done my OCD routine on Sunday for like 2 weeks at least. I still think this is something exclusively reserved for me.
What's the OCD routine for them?
12 hours and a half. Actually, yesterday was not that bad. I was
Let's go
back
over
over to
this
11.
together. So first of all, let's look at our heterozygous individual for both different traits and figure out the allele combinations. I'll do this one on the board here. So again, I like to start with my first one, right? Big R, big S would be a combination. Right, then big R, little s would be a combination. Right, now I've exhausted all my options for my big R. I'm moving on to my little r. So little r, big S is a combination. Or, do a different color here, little r, little s is a combination. Alright, so once I figure out the 4 possible combinations, I just put it across the top of my putted square. Alright, what are the 4 possible combinations of my other individual? Yeah, it's all going to be the same, right? Because even though we'll still do it the same way, the only possible result is still little s, or excuse me, little r, little s. Right? So if we add that in, okay, then we would go through our Punnett square, how we would usually go through it, right? So we combine the first column in row, right, the second column in row, third column in row, fourth column in row. Right? Now if you started doing this, you might have picked up on a trend that because this individual can only give off one genotype in its gametes, all the rows are going to be the same. All right, so if we were to look at how many individuals were red butterflies, how many individuals would that be in this case? right? We could assign our phenotypes here, right, for color. I think I did color, or did I do some fancy work? Oh, I did do body stripes or dots as well. It's very faint there, right? But we could assign our phenotype to our genotype and see that half of these are going to be red, half of these are going to be yellow, right? Again, we would not expect a certain ratio because this is not a true breeding line. Any questions? All right. So we're still talking about a two-factor cross, right? We know there's two different options, either sorting independently or they're being linked, right? Now let's talk about linkage, right? So Mendel's law are true for all genes, right? Law of assortment, law of segregation, but not all traits are going to follow those ratios set down by Mendel. Right? We're going to see two-factor crosses that no longer follow the 9 to 3 to 1 ratio. Right? This being one example of those crosses. All right. So two researchers, Bateson and Punnett, of the Punnett's rare namesake, right, crossed these peas for flower color and pollen shape. Right? So we crossed a purple flower with long pollen with a round flower with round pollen. Again, we can see it's a true breeding line. You can see that in our genotypes here. They got their F1 generation, which were all heterozygous individuals. They all exhibited purple flowers with long pollen. All right, so far so good. We would expect this to happen. But when we self-fertilize two of these individuals, or we cross two of these individuals together, or let one individual self-fertilize, right? So look at the F2 offspring. We got this really weird ratio, right? We got this ratio of 15.6 to 1 to 1.4 to 4.5. Right? That is not a 9 to 3 to 3 to 1 ratio. Right? Because it's different than that 9 to 3 to 3 to 1 ratio, this is telling us that linkage is appearing. Right? This is saying that these genes are linked. And we can see that if we look at what we might expect to see, right? We would expect to see, right, a 9 to 3 to 3 to 1 ratio, right? Which is on the far side of this table. But what we actually observed was this really wonky ratio that was not 9 to 3 to 3 to 1. And we noticed that the genotypes, or I should say the phenotypes, that the parents had, right, in the original line, those true breeding parents, showed up more often than any of the other combinations. Right, so we started with purple flowers with long pollen and round flowers with red flowers with round pollen, and we see that the observed numbers, those are the most majority. Those are in the majority of what we have. Right, so this is telling us that they are not independently assorting. Right, instead we have linkage occurring. Right, because again, the observed ratio is different than what we would expect of the 9 to 3 to 3 to 1. All right, so let's do a quick group review. I want you to show me, I have two different lineages of true breeding pairs. Which one is independently assorting and which one is linked?
First one is
Okay, which means.
The first one is 9 to 3 to 3 to 1. And
So,
then-
yeah. Because a weird ratio.
We did it. Figure it out.
So, that's not out.
Just felt like really they stole my pressure. I was about to say that.
All right, so I've shown you two different possibilities that could result from the true breeding cross here and result in a different ratio for each F2 generation. So which example, the one in blue or the one in green, is showing linkage?
Blue.
Yeah, the one in blue, because it all boils down to the difference in ratios of what we would expect to see and what we actually observe. We would expect to see, with independently assorting, a 9:3 to 3:1 ratio, right? But in blue, what we actually observed was like, if we reduce this down, a 1:7 to 7:1 ratio, right? Which is not 9:3 to 3:1. And because it's not 9:3 to 3:1, we know that this case in blue, these genes must be linked. All right. Any questions? All right, well, let's keep talking about linkages. So oftentimes, organisms are going to have sex chromosomes that will have genes linked to only those chromosomes, right? So this is called sex-linked inheritance, right? So sex chromosomes are found in many species, not necessarily all species, right? And it might have differences depending on which ones we're looking at.
I'm so sorry. I'm just here to pick up Caitlin.
She has already left.
Oh, okay.
Yes.
Thanks.
Yes. So we had a visitor in class visiting our class, if you noticed. Somebody who you didn't recognize earlier. All right, so our sex chromosomes in humans, right, we work on this XY system, right? And males are XY, females are XX. But in other organisms, this varies, right? Other organisms have different classifications of their chromosomes and what makes them, their sex male or female, right? So for example, grasshoppers have this XO system, right? These chickens or birds often have a ZW system, right? So there's variations on this, right? Even within our human chromosomes, there can be different variations of our chromosomes. For example, if we have like an intersex individual. Right, but we're going to stick with this classic XY example and look at how certain genes are linked to these XY chromosomes, right? So, if we're thinking about humans, right, we're thinking about the XY system, right? If you're a male, you have an X and a Y. If you're female, you have two X's. Now, if we look at the size of the chromosomes, right, the X chromosome is actually larger, right? And because it's larger, it carries many more genes, right? There's more genes associated with the X chromosome, right? Simply due to size. So oftentimes when we talk about linkages on sex chromosomes, we often are talking about X-linked genes, simply because it's more common, because the X chromosome is larger. All right, so we would call these X-linked genes, right? They're found on one sex chromosome, but not the other. And for males, right, that only have one X chromosome, there's actually a special term for that, right? It's hemizygous for that particular chromosome, meaning they only have one copy of the chromosome instead of two. And in this image here, we can see the physical size difference between the X and the Y. Now, there are cases of certain traits being linked to the Y chromosome, but by and large, most of them are X-linked simply because of the size of the X chromosome. All right. Let's look at an example. So we're going to look at this example of individuals with hemophilia, right? So that is having some uncontrolled bleeding. If we were to write out the genotypes for these individuals, it's going to look a little bit different than if we were to write out, or how we've been writing out the genotypes previously, because they're linked to our sex chromosomes. All right, so if we look at that first example there, we have a female. It's labeled as a normal female because she does not exhibit or carry the trait for hemophilia. Right, so she's a female, so 2 X's. Got my chalk here. Right, so there's 2 alleles, right, because there's 2 X's. And this female is normal, meaning she does not exhibit this trait, right? And the normal allele is dominant here, so we would have 2 H's. Both superscript above the XY chromosome, or above the X chromosomes in this case. Right, now if we have a female with hemophilia, because it's a recessive disease, she has to have 2 small h's. Right, we see that denoted on their genotype. We can also have a female that does not have the disease, but is a carrier for the disease. Meaning she is heterozygous for that allele. Right, she has 1 dominant allele and 1 recessive allele. Right, so she does not exhibit the trait because the dominant allele is not exhibiting the trait, and that masks the recessive allele. Right? But she does carry the allele for it, and therefore has the possibility of passing it down to her offspring. All right, now if we look at the male, right, we can have a normal male or a male with hemophilia. Right, if we're writing the genotype out for male, we have to include the sex chromosomes, XY. We know this is an X-linked disease, right? So if we look at a male with hemophilia, this male will have a recessive allele, but only on the X, right? We're not going to write anything for the Y because this gene cannot be present on the Y chromosome, right? So we have X, superscript small h, and then Y. All right, so if we cross 2 of these individuals, we're going to cross a female that's a carrier for hemophilia and a normal male. We could set up a Punnett square exactly like we've set up 1 factor Punnett squares before, except this time we have to include our X and Y chromosomes when we lay it all out. All right, best way to do this is to practice. So in this scenario here, we're looking at color blindness, which is a sex-linked trait on the X chromosome. Normal color vision is the dominant trait here. And we're looking at a cross between a female who is colorblind and a male who has regular color vision, right? So let's figure out genotypes, and then put it into the Punnett square to figure out what the offspring would look like.
The only 2 options we can get split would be I or X baby X roll.
Yeah. So all females are going to be normal.
All females are going to be normal because with colorblindness is actually typically
My brother used to think like
No.
certain kinds of
No.
yeah, we told him to stop because he believed it. He was like 13. Yeah.
Really?
He's like, yeah, I get it.
because the man is normal vision.
But like, he was like, no, we're like, dude, that's what it is. Like, that's
Wait,
what are you talking
I
about?
thought the woman carries the abnormal gene.
No, it's a woman who's colorblind.
Oh,
so
okay.
she's not-
Yeah,
Okay, yeah, then all the males are going.
Hold his own.
That's why,
Is
yeah, that's
that-
why
Oh,
colorblindness
yeah.
is
We're like, we're trouble.
Yeah.
at least certain kinds of it. Gray matter, whatever.
Yeah.
Yeah.
I've only ever met one person who's colorblind.
squares, come on here.
Good. Okay. So question: when you have something like this, where you have like both of the males are gonna have regardless, no matter what the male is, they're gonna have it. Is that the same for like something here? Or is it like truly 50-50 on like, oh yeah, you're gonna have it, oh you're gonna have it?
For chance.
Yeah, like so. I mean, obviously it's like 1 and 1, but does it count for like a second trait, or
Yeah, but
is
if you
it
really think
like
about
okay,
it,
if you're male, it's gonna be
we don't know if everyone sees colors the same way.
Is there more than that? That's
They don't. Women see one way.
If it's a
That
that's
I
like
mean,
only
like,
one.
considering that in my family, like,
If it's
I
each
have so
that
much of a better sense of
That's like a
than
thing.
yes, that there's
Yeah,
more.
it's because women see more subtle differences like the wider
Hopefully it
Have you
does.
done one of those like tell the difference between the colors type tests?
Of course.
No, but I've done a lot of the color
When is
blindness
the snow
tests.
starting today?
It says 7, but it's supposed to rain all day, and then it's supposed to freeze. So I don't really know.
Optimal.
I'm leaving campus as soon as I can because I have a commute. So
I did.
How far? Yeah.
I live in Chaska, so
45 or not, you
Okay.
can tell the difference between like the different.
It's a little hefty drive to make every day.
It's one of those tests that
bad.
asks you to
No
rearrange the
traffic.
colors in order.
I rather have 45 minutes with no
I
traffic
got
than
like
20 minutes.
100% on that test.
Yeah.
Mostly because that one is relatively easy. Well,
Yeah.
give that test to
So I think
someone
that's
who does
90% of
nail
people
polish
in
for a
from
living.
wherever they're coming from.
They're never going to fail it ever.
Hi.
All right, so we have to figure out our genotypes of
What
our
do you take?
males and our females here.
Everything but the
So our female, let's see, our female is colorblind. What is going to be her genotype?
Random brother.
Yeah, X little b, right? She is colorblind, which means she can only have the recessive alleles for this trait to show up in her phenotype. All right. What about the male? What is the genotype for the male here who has regular color vision?
That's our man.
Yeah, X, big B, Y. There's nothing on the Y, again, because this gene is only found on the X chromosome. All right, so hopefully you set up your Punnett square, made the correct crosses. Right, what percentage of the male offspring are going to be colorblind? 100%, yeah. Because colorblindness is an X-linked trait, right? This is why it's more common for males to be colorblind than females. But females can be colorblind, right? It's not unheard of, it's just more common in males. All right. This is my note to make sure I check the time and we have plenty of time to keep going before the content check. So let's talk about some irregularities in how phenotypes are expressed, right? So a lot of times, phenotypes and their underlying genotypes way more complicated than what we've been talking about, right? Often there's way more that goes into how a phenotype is expressed than one gene equaling one trait. Okay? So for example, we can have incomplete dominance, where we might cross, in this example, 2 flowers. They both have dominant alleles for different colors, right? So they're either dominant for the white color or dominant for the red color. And when they pass it down to their offspring, so they have one R and one W, right? One specific phenotype isn't winning out over the other. Rather, it's a blend of the phenotypes. So instead of getting white to red, we get pink because of this blended phenotype. So they're both showing incomplete dominance. Right, now if we were to take this F1 generation and cross it to the F2 generation, we would see reappearing the white and the red flowers along with the pink flowers. All right, so it could be a case where we're blending 2 different phenotypes together. That's one possibility. It could be that these individuals, right, still have both dominant alleles. They're both homozygous dominant for their trait. But instead of showing incomplete dominance, where it's blended, right, both traits are going to show up at the same time. So here we have an example with these cows. Right, we have this white cow and this reddish brown cow here. Right, they both have dominant alleles. they are both homozygous dominant. And when we have their offspring, which is one from each allele, capital R, capital W, we see that we get cows that are white with brown spots, right? The brown color isn't going away. The white color isn't going away. Rather, they're showing up together, and in this instance, they're showing up as spots, right? So this would be codominance. If this was incomplete dominance, instead of seeing spotted cows, we would see cows that are like a lighter brown color, right? It would be blended. But we're not seeing the blending, we're seeing both color extremes showing up at the same time. All right, so these are a couple examples of how dominant alleles could be expressed for certain traits in certain species. We also have instances where that we have one gene that influences many traits. Right? And again, so far we've been talking about one trait, one gene. Right? Here we're talking about one gene, multiple traits. And this is known as pleiotropy. Right? So a really good example of this is Abraham Lincoln. He had what's known as Marfan syndrome. Right? That's actually a gene that has a mutation in it. And that caused multiple traits to appear on him. Right? He was abnormally tall, or not abnormally, disproportionately tall, and had really long limbs and fingers. Right? He had an abnormally shaped chest, and he had some heart problems. Right? So just a mutated gene, one singular gene, caused all of those traits. Right? This is also really common. All right, we could look at the opposite of that. All right, so far, we've been looking at qualitative traits when we talked about our pea plants, right? Discrete phenotypes, where either it's yellow or purple flowers, either it's red or green seeds, right? Either you have it or you don't, right? Maybe you have a disease or you don't, or you can roll your tongue or you don't, right? These are all quantitative traits, right? You have it or you don't have it. But there are other traits called quantitative traits instead of qualitative traits. These quantitative traits show a wide spectrum of phenotypes, right? So these are genotypes that are influenced by different genes, multiple genes, or they're influenced by the environment in which they live. Right? So this would be a result in a range of phenotypes. So height, which is what this picture is showing. These individuals are lined up by height. Right? We see a range of heights. Skin color, weight. Right? These are all ranges of phenotypes that can be shown. And like I mentioned, this can be either contributed by multiple genes influencing one trait, or the environment can have an influence. All right, my next slide, we're going to look at multiple genes having an influence on the trait. All right, so we're still talking about quantitative traits here, right? Quantitative traits, we could also call them polygenic traits. Poly meaning many, so many genes influencing one trait, right? And this is an example of having many genes, right? Corn color, right? We could have corn that's on one end of the spectrum, very white, and on the other end of the spectrum, a deep red. And if we look at this 3-factor Punnett square cross here, we see it's actually 3 different genes and how the recessive and dominant alleles are playing a role in those 3 genes that influence the one color trait that it will appear as. Right? So multiple genes influencing one color. Right? If they're all recessive, we get a white kernel. If they're all dominant, we get a very deep red kernel. And then we have a whole range of phenotypes in between. All right, so quantitative traits being influenced by many genes. Yeah?
What's like the biological advantage of having this thing being controlled by multiple genes? It seems to be an extra source of bother. For something as not so significant as corn color.
Yeah. Well, maybe corn color might influence what eats it and then disperses it. I'm thinking the biological significance is just genetic variation. You have the variation so that you can adapt to different areas where some animals might disperse deeper red colors than other areas in the environment where animals might prefer lighter colors. Yeah. All right, so still on the topic of quantitative traits, we have environmental influences. All right. So a really classic example of this is actually rabbits. All right, so this is where exposure to different environments can lead to different traits being expressed. Right? So again, we're talking gene expression, so we're talking the central dogma of biology, DNA to RNA to protein. Right? We're also talking about regulating genes, right? Whether or not these genes are actually going to be expressed, turned on or off, right? The amount being produced. So if we look at this rabbit, right, it's being raised in two different temperatures, warm and cold. A rabbit raised in a warm temperature, right, is actually going to indicate that we're not going to use a gene, right? We're going to inactivate the fur pigmentation gene. So if that's inactivated, we're not going to make any proteins, right? The gene product is not going to be made, and it's going to lead to rabbits with white fur, non-pigmented fur. All right, but if we have this rabbit in a cold environment, the temperature now is indicating that we should activate our fur pigmentation gene, right? So the gene product is made, right? And the result is that this rabbit has pigmented fur. All right, so we can think about this process and what part of this is gene regulation and what part of this is gene expression, right? So what is regulating the gene? Right, well, that's temperature. That's determining whether this gene is going to be inactive or active. Right? And then regulation of the gene, right? It's being made, how much would be our gene expression. Now, why for these rabbits would it be beneficial to have pigmented fur on their ears and their extremities?
That's where they lose heat the most to control their body temperature, and black absorbs more sunlight.
Yeah, exactly. So in this case, it's beneficial for the rabbits because black absorbs a lot more heat. Think about if you walk outside on black tar, like blacktop versus the grass, right? Blacktop is really hot. It absorbs the heat, so it's going to keep this rabbit warmer in these cooler temperatures. All right, let's put these terms into practice here. I want you to tell me which of these traits are qualitative and which are quantitative. And I'll give you about a minute here.
Which two are qualitative? It depends on how you would consider it. I would consider it.
Presence of trait is definitely quantitative.
Yeah, I would consider it qualitative. It's just that Rubel is a special exception that just exists.
You mean the blood type?
Yeah.
Yeah, no. I think it's
I'm blood type O.
I actually don't know my blood type.
Oh, that's unusual.
I know.
I don't think it is. I think a lot of people don't know my blood type. I only know it because I've had my blood cancer many times.
Yeah.
Oh, me too. I have blood type B. Although, because my mom is O, and O is like the rare.
Yeah.
But AB is also rare.
Yeah.
No, I meant that O is the one with none of the.
Yeah.
Yeah. So like, if you have two people with O, it's like you can only get like a child with type O.
Yeah. So if your parents have type O and you don't.
No, my mom has type O, my dad has type B.
Yeah, but if both your parents have type O and you don't.
Oh, that's although there is one situation I've seen on the news where that happens, and this child is actually
All right.
is actually their
So I've given you 4 different traits here. I want to know if they're quantitative or qualitative. What about this first one, skin tone?
one developing, and so that.
Let's do that again. What about skin tone?
That does happen.
Quantitative. All right. Yes, it is quantitative. What about the body size of dogs? Quantitative. And there's a range. What about the your blood type?
Qualitative.
Qualitative. You're either one or the other or the other or the other in this case, right? You can't be A, B, and partially O, right? What about presence of a mane? Yeah, qualitative. You either have it or you don't. And presence of a mane, probably an X-linked trait, or excuse me, a Y-linked trait, right? Because it only shows up in males. All right, so the next few slides actually relate to our contents check. But we want to talk about how we might look at traits, particularly in humans, right? So in humans, it's very unethical to run experiments where we force humans to mate that have nice traits to produce the most ideal offspring, right? Very unethical. That should not be happening, ever, right? So because we can't ethically do that, right, like we can with other animals like mice and rabbits, right, we instead look at our genetic history, right? And oftentimes we look at a pedigree, all right? So a pedigree is basically a record of your ancestry, paying particular attention to a trait, right? So if we're looking at a pedigree, right, it kind of looks like a family tree because it basically is, right? Females will always be a circle, males will always be a square. If the trait is expressed by the individual, right, the square or the circle will be filled in with a color, right? This color being blue in this case. If we know that the individual is a carrier, right, it would be half filled in, meaning they don't express the trait, but we know that this individual has the allele for this trait. Now, if they're unaffected, right, they're not showing this particular trait we're interested in, right, it's just going to be an open circle or an open square. All right, so by looking at pedigrees, we can look at these traits going back many generations in a family. So let's look at some examples of this. All right, we're going to look at this trait, hemophilia, which we already know is an X-linked disease, right? We went through an example when we talked about X-linked genes here. And if we look at this royal family, right, we can look back in their pedigree to see that Queen Victoria was a carrier of the disease who passed it along to her daughter Alice, who is also a carrier of the disease, who passed it down to their daughter Alexandra, who is also the carrier of the disease. But in this case, they passed it down to their son Alexis, who actually exhibited this disease. Hence why his square is fully drawn in, right? Now, this is an X-linked disease, and we can see that because in the females, they're all carriers, right? Now, they could exhibit this as well. But in males, right, it's definitely showing up, right? If one of the moms, or one of the mom, right, passed it down. All right, so again, carrier means that you have the allele, right, but you're not exhibiting the trait because it's masked by the dominant allele. All right, so this is an example of an X-linked disease and how you would read a pedigree. Right? There are also examples where they might be sex-linked, or they might be autosomal linked. So autosomal cells are or autosomal chromosomes are all the other chromosomes beyond our sex chromosomes. Right? And in this case, Huntington's disease is an autosomal linked disease, so it's on our autosomal chromosomes, right? And it happens to be a dominant disease, meaning if you have the dominant allele, you express this disease, right? And so we see in our pedigree here It's showing up much more often. It's in males and females relatively equally, right? So this is an example of a dominant disease that is on an autosomal chromosome. All right, one thing about pedigrees that we should talk about is that there are problems with it, right? It's not as simplistic, right? There's always going to be some caveats. For example, just because a trait runs in your family does not mean it's genetic, right? Maybe everyone in your family grew up in the same city, all drank the same water, had the same environment that they grew up in, right? Maybe it's environmentally influenced, right? Maybe it's a nutritional deficiency that runs within your family or within the area that you live, exposure to an environmental toxin, right? It could even be things like language and dialect, right? Or even sports team allegiances. Lots of times, families all have the same sports team allegiance. It's not genetic, right? It's just what you decided to pledge your allegiance to, right? We're also making the assumption with pedigrees, though, that each trait is controlled by one gene, right? And we know that's not the case. In fact, in many cases in biology, this is not the case. So we have to make this major assumption, right? For example, you might see pedigrees of polydactyly in humans. Polydactyly is where you have an extra digit on your hand or your toes, right? It's caused by mutations in multiple genes, right? So if we were to look at a pedigree of polydactyly, we'd be making an assumption that's controlled by a single gene, when in fact it's not, right? So there are problems with pedigrees, right? And if you've done your eugenics assignment, you might also see how pedigrees can be used unethically as well.